welcome to the 14th talk in the webinar series on genetics and evolution organized by the Journal of Genetics. I'm very happy to welcome Professor Volker Loeschcke. He works at the Department of Bioscience, Section of Genetics, Ecology and Evolution, Aarhus University. Professor Loeschcke was born in Göttingen, Germany, and he had his education from the Free University of Berlin. He did his master's in mathematics, went on to do a PhD in genetics and a Doctor of Science in population biology. He has held various positions. He started out as a research assistant in Berlin, then a senior lecturer, reader, research professor, and a full professor at Aarhus University. Professor Loeschcke has been very productive, very versatile as far as research goes. He has very wide-ranging research interests. He's worked on evolutionary genetics, environmental stress response, heat shock proteins, inbreeding, adaptation to extreme environments, thermal adaptation, phenotypic plasticity, epigenetics and transgenerational effects, adaptation to climate change, aging, biodiversity, habitat fragmentation, conservation genetics. The list is very long. Professor Loeschcke and his group have pioneered the study of thermal adaptation and environmental stress resistance, combining evolutionary genetics and ecophysiology using the Drosophila model and employing state-of-the-art molecular techniques. He's published extensively on inbreeding and contributed to studies of aging and the role of diet, and has been active in promoting conservation genetics. Professor Loeschcke has published more than 380 papers and co-edited 7 books, and he is among the 20 most cited European evolutionary biologists. He has received many honors. I'll mention just 2. In 1996, he was elected to the Royal Danish Academy of Sciences and Letters. In 2017, he was awarded the Order of Dannebrog by the Danish Royal House. Today, he will be speaking about plastic and evolutionary responses to environmental stress. Professor Loeschcke.
Thank you, Professor Prasad, for the invitation to speak to you and for the friendly words to get us started. I feel honored to talk to you about my work. And this work is more or less done over the last 20 years on stress responses. And it has been done together with a lot of former PhD students and postdocs, but also a number of colleagues, which are mentioned here on the slide. And I have also to thank you, the funding organizations at the Danish Research Council, so the Carlsberg Foundation, and a couple of other foundations. Let me start to ask, what is stress? What is environmental stress? And stress can be desiccation, starvation, competition, predation, alcohol, pesticides, hard mental work, also inbreeding. But to have a definition that can be used in an environmental context, it is nice to have a relation to fitness. So according to Hoffman and Parsons, I define environmental stress as any environmental change that drastically reduces fitness of an organism and stimulates physiological, metabolic, or behavioral adjustments in body functions. If we take temperature as a stressor and look at fitness components, viability and fertility here, we can see that in the orange range of the temperature scale, fitness is drastically reduced. If we take a real example from Drosophila, we can see that below 14 centigrades or below 12 centigrades and above 32 centigrades, viability is drastically reduced, so we have environmental stress. And if we take this into this information, into this figure, we can say above 32 and below 10 centigrade, organisms cannot complete the whole life cycle, and about below 12 centigrades and above 30 centigrades, males get sterile. But in nature, temperatures are not constant, so we have to also introduce a time dimension. For example, here, Drosophila busatii is exposed each day to a couple of hours of 40 centigrades or even above in summer. So we have to introduce also a time dimension to this stress perceived by an organism, which then is determined by temperature times exposure time. If we are in this situation, we can discuss possible responses to reduce the detrimental impact of thermal stress, and these responses include plastic responses induced by acclimation or hardening that occur within a generation, or the long-term across-generation response of evolutionary adaptation. There is also a possibility of behavioral avoidance that also can evolve, but I will not talk much about this here. What I will do in the following is to compare costs and benefits of evolutionary versus plastic responses to environmental stress. I want to identify mechanisms behind the stress response and behind variation in candidate genes, metabolites, and proteins involved. And I want also test laboratory-generated results for their ecological relevance under field conditions, because if these lab results do not make sense under natural conditions, they are not of much use. I also want to test for ecological and phylogenetic constraints for thermal adaptation. That means if certain branches of a phylogeny are somehow fixed and frozen, and adaptation to changing conditions is difficult. At the very end, I just will also mention a few ongoing work and the results from the microbiome. I used the model organism Drosophila, which you all know. It has a couple of advantages, but it has also the advantage that you have cosmopolitan species and you have also a lot of specialist species. On top of that, Drosophila is molecularly very well described. Let me start out again with some working definitions for hardening and acclimation as non-lethal exposures to environmental conditions that subsequently increase or just change tolerance to more extreme conditions of the same kind. If we, for example, look at the critical thermal minimum, CT-min, as a function of different acclimation temperatures, we see that on a level from 12 to 30 degrees change of thermal acclimation temperature, that is, means the temperature from egg to adult, we get a 6 centigrade increase or decrease in CT-min. So this is an enormous effect that comes from acclimation. If we look at 13 other Drosophila species, we get similar responses. So apparently this effect of developmental acclimation is very general, and you can see slopes of these regressions are very similar. If we instead look at critical thermal maximum, CT max, we see that for the same range of developmental temperatures, the change in CT max is only 2.5 centigrade in Drosophila melanogaster. So it is a kind of lesser response to acclimation here. If we look now at 13 other species that we just had before, we can see that actually for CT max, there is lots of variation. There is no one general response. Some of these figures show a decreasing slope, and some do not show any response. If we go out in nature and look at natural acclimation or acclimatization, which is called under natural conditions, we here see for 4 species that we put in outdoor cages that the longer the acclimation or acclimatization works, the lower is the critical thermal minimum here on the y-axis, and we continued from mid-December to early January in 3 of the species, and for Drosophila melanogaster we went one more step, and additionally you get some improvement in cold resistance, which here is shown for males and females. If we look instead on the hot side of the temperature and look at improvement of heat resistance measured as survival in percent after heat hardening treatment of 1 hour at 37 degrees, we see that the resistance to heat with about 30% survival for non-acclimated flies goes up to 75 or so % of acclimated flies, and the response thereafter gets cancelled out after 2 to 3 days. Look at the mechanism behind this heat hardening response. We see it is what is called the heat shock response. It is the induction of heat shock proteins that protect cells from damage by refolding denaturated proteins and by turning damaged proteins to the intracellular degradation system, a kind of housekeeping function. These heat shock proteins exist in more or less all organisms one has looked for, and in Drosophila here to the right, you can see that the heat shock protein 7G is the major heat shock protein which shows the most induction. And these proteins are measured in kilodalton. If we instead look at the expression levels of HSP70 following a 1 hour at 37 degree hardening treatment, we see that levels of HSP70 immediately are upregulated more than 100 times, and then these levels gradually go down to the basal level after 2 to 3 days. Different stressors induce HSP70 in Drosophila, not only heat, but also cold, high density, inbreeding, inflammation, and parasites. And you can say the role of HSP70 is that it reflects levels of stress perceived by an organism and thus can be used as an unspecific stress marker. For example, if you look at HSP expression after a heat hardening treatment for flies that originate from different latitudes, and that is from different thermal zones. Flies that come from high latitude, their temperatures are usually lower, show much more of an emergency response, that is, they show more of HSP70 expression than flies that come from lower latitudes where temperatures usually are warmer. And you can see a similar response along altitudes where you also have different temperatures. The higher you come, the colder temperatures get, and the more emergency expression of HSP70 you show. If you have such a nice scenario where you can induce heat shock proteins to track environmental change, it is obvious to ask if there are any costs connected to the induction of HSP expression in Drosophila. And here, where we look at the number of offspring produced after 1 to 2, 3 to 4, and so on days, and we can see that for the first 2 days, offspring number of hardened flies is only 50% of the no hardened flies. So apparently there is a kind of strong reduction in reproductivity, but it only varies 1 to 2 days, and after 4 to 5 days, the hardened flies show the same productivity as the non-hardened flies. You can also ask what happens if you acclimate for one specific trait, what happens in response to other traits. So is there any cross-resistance? Are there any trade-offs? And we did an experiment where we acclimated for cold, for heat, for desiccation, and for starvation, and then to the left, the column to the left, and then tested for cold, heat, desiccation, and starvation resistance. And first, in the diagonal, we see plus signs, because if you acclimated for cold, you got also higher cold resistance. If you acclimated for heat, you got higher heat resistance, and so on. But when you then also look at the other signs, you see that, for example, if you acclimate for heat, you get no effect on cold resistance, you get a positive effect on heat resistance as expected, but you get also a negative effect on desiccation and starvation resistance, and in general, except the diagonal, you have more minus signs than plus signs. You have that means you have a lot of negative responses to acclimation. You can also look at acclimation effects in the wild. Do laboratory results reflect what happens under semi-natural conditions? And for studying that, we introduced a kind of release recapture experiment where we dusted flies with fluorescent colors and then released them at the release point and had around buckets with food with mashed bananas standing, and then we considered the ability to locate resources as a kind of fitness proxy. And as in natural situations, fruits dry out, this looked to us for a kind of natural situation. And what did we get? If we, for example, take the squared here, 15 centigrade developmental acclimation, we see that if temperatures are cold, at 12 or 13 centigrade, the flies that got acclimated at 15 centigrade are 100 times as often found in the buckets than flies that were not acclimated. So we find an enormous effect of this developmental acclimation for flies to locate resources when temperatures are low. And the same flies showed 30 times negative effect as compared to controls, which are normalized at the 0 line, or as we have a logarithmic scale here, as the 1 line. So we get enormous effects under natural or semi-natural conditions by releasing flies at different temperatures depending how they got acclimated. If we sum up the effects of hardening and acclimation, we can say hardening and acclimation treatments may have large beneficial effects if hardening or acclimation and stress type are similar, but otherwise they may have strong negative side effects. That means strong costs with respect to traits other than those acclimated for. So thermal adaptation based on plasticity can be very costly. Let me shortly also show you other consequences of heat hardening of 1 hour at 37 centigrades. We get temporary changes in gene and protein expression and metabolic profile. For example, if we heat harden at 37 degrees for 1 hour and look at the heat map, that is at the gene expression just before the heat hardening treatment, that means at -1, and compared to 4 hours after the heat hardening treatment, we see that these two values resemble each other most, but we can also see that there occur changes in the heat map, in the gene expression, up to 64 hours after the heat hardening treatment. Just a link to the metabolic profile. The heat hardening treatment shows that the time response of the gene expression and the metabolic profile are similar. Both peak after 1 to 2 hours after treatment, and the effect disappears after 4 to 8 hours here, where you come back to basic levels. Let me now go over and look at evolutionary adaptation and compare the results from evolutionary adaptation to those from acclimation. And we did a large selection experiment where we selected to improve tolerance to different environmental stresses as alternatives to plastic responses, and look at if we do this with lower costs. So we selected for 3 different heat resistance traits: heat resistance in a water bath, survival at constant 30 degrees, and heat knockdown resistance in a knockdown tube. And further, we selected for cold resistance, for starvation resistance, for desiccation resistance. And we also looked at longevity and selected for longevity because it has in other studies shown some resemblance to effects of stress selection. What did we get? We get that first, let's have a look, for example, on the figure. Sorry, the cursor has a slow working. Here on this figure, when we tested for on the upper left for cold resistance, we could see that flies that were cold selected, CS, had the highest cold resistance. This means selection was successful. And the same is true if we look at heat resistance. Flies HS, heat selected, that were selected for improved heat resistance, shows the highest heat resistance. And if we take the two figures below, flies selected for starvation resistance, S starvation selected, showed the best performance when testing for starvation resistance, and flies selected for desiccation resistance, DS, performed best when tested for desiccation resistance. So we got a kind of significant improvement of stress resistance for that trait that was selected for, but we got also that, for example, here, that other knockdown selected and desiccation selected flies had higher heat shock resistance than the controls that were normalized to the zero line here. So we got a lot of positive correlated responses, at least in the four subfigures to the left. When we tested for heat knockdown resistance and longevity, we did not get so clear-cut results. For longevity, not, as we only had 7 generations of selection as compared to the 20 generations of the other treatments, because it just takes extra time to select for longevity. And for heat knockdown resistance, apparently this trait is just a muscle that does not work anymore above certain temperatures. So in a way, we got clear-cut correlated responses, beneficial correlated responses, which somehow indicates that there is a kind of general stress response. If you select for one stress resistant trait, it usually also improves other stress resistant traits. If we look at some other side effects, we can see that developmental time gets a little longer after selecting for stress resistance, except for the heat selected lines that also, with respect to other traits, behave slightly different. If we look at body size, here measured as thorax length, we find that there was a significant response in the longevity selected lines, but not much response, except that there was a tendency that flies got a little smaller when you selected for stress resistance. Let me just look how far I have come here. Let me also look at some omics performed on the selection lines and a few results under natural conditions. And first, gene expression. We can see that the different replicates, we show 3 replicates only because these gene expression studies are quite expensive. We can see that these 3 replicates of the different selection lines nicely clustered together. Here's the desiccation selected, here's the starvation selected, here's the longevity selected, and so on. So from the 260 genes that showed significant changes in expression, we could see some indication of a general stress response. And if we, for example, looked at some of the specific selection lines, we could identify candidate genes, for example, among the phototransduction genes. The majority of genes of these genes was upregulated, and only a minority of genes were unchanged. If we looked at the proteome, we could see that the number of significantly expressed, differentially expressed proteins was, if you did pairwise comparison, for example, of desiccation versus constant 30 degrees or desiccation versus cold selected, that these changes 15 or 7 or 10 were in the same direction, which also indicates to a certain degree that you get a kind of, that you have a general stress response. Changes are for all selection regimes in the same direction. And again, we have one difference, that we get opposite direction changes if you compare with heat selected lines, where you get this few shared significant proteins that respond in opposite directions. And here also for the proteins, you see that the 11 heat-shock proteins that are shown here all are somehow involved in changed expression levels of the proteome. So if I sum up this last result, I can say selection for stress-resistant traits usually leads to positive correlated responses in other stress-resistant traits without any obvious larger costs. So in a way, you could say they all point to a general stress response that we also saw on the phenotypic level. Selection for stress resistance leaves strong signatures on gene expression, metabolic profile, and the proteome for each selection regime. It shows some but not very strong overlap in responses between the different levels of biological organization, phenotype, transcriptomics, metabolomics, proteomics, and later genomics. Indicates strongly the existence of a general stress response based on data from the proteome, transcriptome, and metabolome, and demonstrates an important role of the heat shock proteins for the general stress response. And it also demonstrates that heat selected lines are most different from the other selection lines. If I now go over to the sequencing results, the genome sequencing after 21 and 31 generations of selection for increased stress resistance, where we again have here all 5 replicates, and now I find the cursor, where we here have the 5 unselected controls, the 5 heat shock resistant selected lines, the 5 starvation selected lines, and so on, and where we look at these lines at the sequence after 45 or 65 generations, which means after 21 or 31 generations of selection, because we have 1 generation without selection between selected generations, and because we started selection only after 4 generations to mix all the populations in the beginning. When we look at the results, I show here the Venn diagrams of the for polymorphic sites of interest, PSIs, that are shared among the selection regimes. We can go in detail in this kind of mixtures and see the genes that overlap between the different selection regimes, and go more in detail to find interesting genes. And if we look at inferred evolution with bootstrap values, we can see these maximum likely trees here, and we can see that the heat selected lines with a 100 bootstrap value in a way groups for itself, and here we have the other different selection lines. So in a way you can say heat selection again shows different results. So if we summarize the results from the genomics or from the sequencing, we get more than 100,000 SNPs comparing unselected controls with selection lines, both encoding and non-coding sequences with highest density and promoter regions. And we get highly stressor-specific responses involving broad ranges of functionalities, including molecular chaperoning by heat-sheck proteins. And surprisingly, only 15% of the SNPs were shared by at least 2 selection regimes, while less than 1% diverged in opposite directions. That means we almost got no trade-offs here. And to whom this is interesting, we also got a decrease in the transposable element frequency. Let us go back to nature and test if laboratory results from artificial selection for environmental stress resistance reflects fitness under natural conditions. And when we tested for the selection lines that were selected for heat and cold resistance out in nature, we can see here the leftovers of the fluorescent dust and the wilds where we transported the flies to this nice Australian scenery. We got a clear effect that yes, at cold temperatures we caught many more cold selected lines, and at hot temperatures we caught many more of the heat selected lines. So what we did in the lab also showed to be true under natural conditions. Now ask if phylogeny constrains thermal adaptation, because Drosophila species may be phylogenetically constrained that on a branch of the phylogeny it is difficult to change resistances because their forefathers in a way determine and constrain this results. So to test this kind of phylogenetic inertia, we did a large stress resistance experiment where we tested 96 Drosophila species for their critical thermal minimum and for the critical thermal maximum by putting flies in this small kind of glass vials in an aquarium where the water was steered around by different pumps. And we started from 20 centigrade and either were ramping up to or decreased temperature ramping down by 0.1, if I take the cursor, then it always, by 0.1 centigrade per minute. So we got a slow increase or a slow decrease, and this 0.1 is a kind of arbitrary change, but it is often used in the literature and considered the ecological most relevant change. If we did this, we got this kind of tree where we have here where we have in the right column the 96 species and the conditions where they were caught, and then we see how these are connected to each other, and generally they behave, they show some unflexibility with respect to changing cold resistance values. So we found some phylogenetic inertia, so this species could not just easily become more cold or less cold tolerant, as some of this cold tolerance was determined by the phylogeny, by where the species came from. Without going in details, you can look at the references that are on each figure. We did the same for heat tolerance and also find some indication of phylogenetic inertia, which you could say means that species are not optimally adapted to the local conditions. Because of this inertia, they could not just change easily. You could also ask now how strong are correlations between thermal performance and the climate of origin, and how well are species adapted to the thermal conditions they live under. And if we look at the CT min values as a function of average minimum temperatures where the flies are caught, we have here the 96 species, and we get a kind of nice linear regression with a R-square value of 0.56. So yes, there is a clear correlation between average minimum temperature at the collection site and the CT min. But if we look at heat resistance of the same 96 flies and compare the average thermal maximum temperature with the CT max, we see that there is no correlation. Oops, sorry, I have to again find the cursor. We see that there is no correlation whatsoever. The R-square value is 0.03, which is surprising, but which also shows that there is a constraint to adaptation on the heat side. Now I somehow got, if we, just to see, go back, this figure we had before, and now we looked instead of annual precipitation, instead of average heat thermal means, and we found that CT max and annual precipitation correlated with a kind of R-square value of 0.149. So there was some significant correlation, but not a very strong correlation. But if you only looked at dry environments, this correlation was bigger. So you could say, as summing up from the last part, heat and cold tolerance are phylogenetic constraints, and thus current resistances may not be optimal. If environments change, it will take time to track these environmental changes. And again, we can also say heat resistance is not strongly correlated with average maximum temperatures at the origin of populations, at least under humid conditions. Let me finish up with just a little data, a few comments on the microbiome. We currently work on the data from the 16S RNA study on the microbiota community, and we find for our selection lines that there is a very clear-cut pattern in the microbial community of the different selection lines. And if we go in details here only in a kind of overview slide, we have here to the left the 25 most frequent bacteria species of phyla, and we have here up this code selected, desiccation selected, heat selected, knockdown selected, and so on lines. And we can see that usually the 5 replicates behave very nice in the same direction. Here you can see again that the heat selected do not have this bacteria, but have instead some other bacteria. So there is a kind of clear difference and a clear grouping. I will not go in more detail as this work is under work, and we are still analyzing the data. So I think my time has gone, so I will stop here and once more say that this work was done in collaboration with Jesper Sørensen, Thorsten Christensen, Pendelisaro, Masco, Tommaso Menenti, that were all former PhD students under my supervision, Pavel Michalak now in the United States, Oleg Bublia in Berlin now, Ari Hoffman as a colleague, and one of Ari's former students, Vanessa Kellerman, and a few more, and it was funded by many of the Danish research foundations that support research. So I thank you for your attention, and I'm happy to answer questions.
Professor Loeschcke, here is the first question from Professor Amitabh Joshi. Were all 13 species of Drosophila used for looking at CT min and CT max after acclimatization from temperate regions, or were there also tropical species?
The species were, we took all species that we could get hold of. So, and we got some species also from stock centers, and then tried to get the origin of these species as those where they usually are found. We took the center of the distributional range. So yes, there were also tropical species, there were subtropical species. We took all species we got hold of.
Okay. Second question is, if hardening is done for two stresses simultaneously, does that alter the trade-offs in subsequent tolerance to various stresses?
Very interesting question. Yes, we played a little bit around with the mixture of hardening treatments, but we have not published anything of that. So that the multiple stresses and its effects could be very interesting for follow-up study. Yes.
Okay. It is strange that many stress selection regimes increase development time, but did not seem to increase thorax length. Do you have any comments, given that typically longer development correlates with larger size?
I think you could say that I just try to get back that thorax length was not a very clear-cut result, but you had some indication of lice being smaller. Developmental time, you could say the heat-selected lines were different, but in this business, you could expect that the flies that use more time for development have also more time to accumulate fat, but this is not always true, or the correlation is not very high. But it's an interesting thought, yes.
Okay. Next question, was the base population for all the stress selection lines obtained by pooling many lines? If so, 4 generations before starting selection could be too short a time for linkage disequilibrium to decay, and this could cause artificial correlated response to selection, especially in the short term, that is about 0 to 20 generations or so of selection.
Yes, this is a fair critique, and I'm aware of that you should have more generations to get rid of linkage disequilibria. But yes, some were mixed, and that is, but they were also in part among the mixtures combined. We took the first 4 generations of these lines without any mixture, but before some of them had been mixed, so there was not new material involved. But fair enough, it's a valid critique. Yes.
Okay. So another question is, a large number of these selective agents, especially thermal selection, quite often acts on things like cuticular hydrocarbons and all that. So this could, at least in some other selection regimes, this does seem to have an effect on how males and females interact and reproductive behavior. Did you see any evidence of that? Did you look at for these kinds of traits in your selection regimes?
Not look at this, but it would be clearly worthwhile to do it. Yes.
Okay. I don't think there are any more questions. So I would like to touch on comment from Mr. Amitabh Joshi, who says, very nice talk, Dr. Loeschcke. Very good to see you again too. So,
okay. Yes, Amitabh, hello.
I would like to thank Professor Loeschcke for a very nice talk. Thank you. And I'd like to thank all of you for joining us.